think other than the comprehensive final exam, that will definitely be the hardest exam you have, so don't give up hope. Have faith in yourselves, okay? And in addition, folks, a textbook publisher had asked if we might consider reviewing one of their micro textbooks. So I'm going to be brainstorming a way that we'll be able to do that this weekend. So if people were asking if there might be opportunity for more bonus points other than the total of 20 bonus points for the extra credit poster. So I'll be brainstorming that this weekend. Okay, and some of your colleagues have given me some good ideas already about that. Folks, before we get started, I don't know if any of you saw the article. This is the Sacramento Bee, Sunday, October 28th. And the headline, this is on page 1B, is Sacramento County finances STD treatment at community colleges. So Saxon College and Cosumnes River College, starting November 1st, our students will be able to get free sexually transmitted infection screening and treatment, which is awesome, guys, because the statistics are just heartbreaking. So these are the statistics printed in the paper, guys. So from 2013 to 2017, there have been big increases in sexually transmitted infections. And the one they reported, you guys, these are all bacterial, and that means that, if they're diagnosed early, antibiotic therapy will take care of these. So you guys, this is horrible. Syphilis, an increase of 136%, right? That's, oh my gosh. And then gonorrhea, an increase of 50%, chlamydia, an increase in 23%. So these are really disturbing statistics, you guys. And not only are we worried about the folks that are infected, but if we have a pregnant mom, all three of these can be transmitted to the baby. And we're always worried, of course, with babies becoming infected because their immune system isn't fully developed. So folks, I thought when I saw this, I was like, wow, kind of like what's going on? So one thought was, you guys, that this might be another extra credit project and maybe more of a report. And my thought was if you all can kind of like brainstorm, what do you think's going on? Why in this day of YouTube and video, we should be well educated. What do you think's going on that we're seeing this increase in transmission of STI, sexually transmitted infections? And furthermore, you guys, I would like you just to brainstorm, use your imagination, in your report, come up with possible solutions, right? Like, well, what if we did this? What if we did that, right? Because this, is, well, for everybody, especially those of you going into some aspect of medicine, this is really significant, right? And we know, you guys, with the gonorrhea and chlamydia, that chronic infections that are left untreated, remember, you guys, it can lead to ectopic pregnancies, it can lead to pelvic inflammatory disease. In some cases, it leads to infertility, right? And syphilis, you guys, is horrible. Left untreated, it can invade bone, and in really horrible cases, it can invade the central nervous system. And sometimes, you guys, once the damage is done with syphilis, even if you treat with antibiotics, the damage is permanent. And the syphilis can cross the placenta, right? So this can cause horrible congenital infections. So again, you guys, thinking of, this wouldn't be a poster, it would be an actual report. Like, for you to maybe do a little bit of research, see, what the thoughts are on why we're seeing these increases in STIs, and then brainstorm possible solutions, right? Like, say you're in charge of sexually transmitted infections in Sacramento County. What would be some of your proposals, right, to get this under control? The other thing, you guys, that really troubles me about this is what aren't they reporting? They're not reporting HIV, right? So, so what's happening with HIV infections, right? What's happening with other viral sexually transmitted infections like herpes viruses, which we will be talking about, HPVs, human papillomavirus, some of which can increase our risk for cancer, right? So to me, it's almost like this is just telling us that the problem's probably even bigger. Okay, so you guys, I'll try to write up a description and post it on Canvas, all right? Because this, I think, is a really important topic for all of us, yeah. Okay. So folks, our plan we're still on the Introduction to Viruses PowerPoint. And folks, I think last time we were talking about the structure of viruses. And yeah, so is this about right, you think, where we left off last time? Talking about how we classify viruses. So you already know we classify viruses based on whether they use DNA or RNA as their genetic information. And we know that we're always a little bit worried about RNA viruses. Why is that, you guys? Why are we a little bit more worried about RNA viruses? High mutation rate. And if on an exam I asked you, why is it that RNA viruses have higher mutation rates than cells or DNA viruses, what would you tell me?
No proofreading.
Yeah, but and what doesn't proofread, you guys? What is it that doesn't proofread? The RNA polymerase, right? Doesn't edit, doesn't proofread, so we have that much higher mutation rate. Good. And then furthermore, you guys, we're gonna classify viruses based on whether they have an envelope or not. So have we gotten to that yet, you guys, explaining what an envelope is? Sorry, okay, we'll just pretend like we did it. Okay, so you guys, this is a rather old falling apart model of a naked virus. So what's this outside coat called? The capsid, good, and it's made out of proteins, right? Okay. And you guys, have we talked about adhesins? Yeah. Okay, good. So you guys, if a virus, if the outermost layer of a virus is this protein capsid, we call it a naked virus, okay? So have we talked about adhesins, folks, the role of viral adhesins? And we use this cool, okay, so you guys, so we can see here in this cool poster that a previous student did. Here are our viruses. Here's our host cell, say a human cell. And these little balls, you guys, these are host cell surface receptors, molecules on the surface of the cell. For the virus to invade the cell and start replication, it has to first attach. And so this is brilliant. These little golf tees, these are the viral adhesins. They have to bind to the host cell surface receptors before the virus can get inside. So the question is, you guys, if we have a naked virus, where will the viral adhesins be? On the capsid, right? And this is actually significant, you guys, because since the capsid is made of protein and the adhesins are anchored in the capsid, this means that naked viruses are going to remain infectious for relatively long periods of time once they're shed from the host. Right? So that's bad news for us humans if we're dealing with a naked virus that's causing an outbreak of infectious disease, okay? In contrast, folks, the second type of virus is a virus that steals some host cell membrane before it leaves the cell. And it could be cytoplasmic membrane, nuclear membrane, endoplasmic reticulum membrane, right? You guys, I know this is goofy, but let's pretend this is the stolen host cell membrane, right? And the virus is escaping. So now what kind of a virus do we have? An envelope virus, right? But you guys, where must the adhesins be? Would it make sense to have the adhesins in an envelope virus? Would it make sense for them to be attached to the capsid? No, right? Because they won't be able to bind the host cell. So you guys, before an envelope virus steals our host cell membranes, what must it do to the membrane? Insert the viral adhesins, right? So viruses can modify our cell membranes. And very importantly, if we're going to have an envelope virus, the virus has to insert its proteins, its protein adhesins into the cell membrane before they steal it, right? Because the adhesins have to be with the envelope. And you guys, this is also significant because remember our model of cell membranes, fluid mosaic model, an ocean of phospholipids with proteins floating in that ocean, the consistency of olive oil. So you guys, do you think an envelope is very tough and resistant? Think of olive oil. No, it's very delicate, very fragile, right? So you guys, the good news for us is that if we're dealing with envelope viruses in some kind of outbreak, envelope viruses will rapidly become inactivated in the environment once they're shed from their human host. And that's because anything that damages the envelope, even just drying, soap and water, right? UV, anything that damages a membrane means the adhesins can't function, right? So the good news for us is envelope viruses don't remain infectious in the environment for long. So you guys, let's just take a look at our table here. These are some of the viruses. Some of these we'll be talking about in more detail later. Okay. So this table, again, you guys, is just an example of how we classify viruses according to genetic information and according to whether they have an envelope on. So you guys, we will later be talking a little bit more about a naked DNA virus. HPV stands for human papillomaviruses, the so-called wart viruses. So we do want to remember, because they're naked, they're going to remain infectious in the environment for longer periods of time. And then you guys, a naked RNA virus is poliovirus. This virus is shed in feces, right? So wherever the feces goes, the virus goes. So fecal contaminated food or water is of huge concern, right? Because poliovirus might be present. And again, you guys, because it's naked, that means it's going to remain infectious for long periods of time in feces, in contaminated food or water. And then, folks, we'll briefly be talking about two envelope viruses, the herpes viruses and the hepatitis B viruses. And then two envelope RNA viruses, you guys, we'll talk about in greater detail, influenza virus and HIV. And to me, this is like the only good news about HIV is luckily it's an envelope virus. So that means if, like for example, you guys, if I have HIV, let's say I cut my finger and a droplet of blood falls on the floor or on the desk here, as soon as the blood dries, the envelope is going to be damaged, and that HIV is no longer infectious. So this is really important, you guys, that you understand kind of like the ability of a virus to remain infectious in the environment. When HIV was first discovered, a lot of people was like, I don't even want to be in the same room with somebody that has HIV because, what if they touch the doorknob and then I touch it and I'm going to get infected? And it's just ridiculous, you guys, right? So the good news is with HIV, it's envelope. It's not going to remain infectious in the environment for long at all. Yeah.
So is an envelope virus essentially a naked virus before it steals the membrane?
Yeah, right.
So
Right. So if you take a naked virus and you wrap it in stolen host cell membrane, that's an envelope virus.
So why can't it just be a naked virus once it's in the environment after its membrane?
Because the way they evolve, where are their adhesins located? Yeah, so their replication cycle, they're in an envelope virus, the adhesins are always going to be in a host cell membrane, and they've just evolved so their adhesins aren't on the capsid. So you're right. I mean, Martin's right. So let's say this is HIV, right? Shed into the environment. The envelope is damaged. So why isn't this infectious? It's because it doesn't have the adhesins. Yeah. So like I said, lucky for us.
Well, how is it infectious before it steals the membrane?
Because the infectious form is this form. So like right now, you guys, if let's say maybe I was a healthcare professional, I was taking blood from one of my HIV patients. The HIV circulating in the blood is this form, the envelope with the adhesins here.
Mm-hmm.
Okay, so I do the needle stick, right? I get some blood with HIV in there. This is the form that HIV is circulating in my body. Then it binds to my macrophages or T helper lymphocytes. Then it gets its genetic information within my cell. It replicates and it escapes, stealing more host cell membrane. So for an envelope virus to be infectious, it has to have an intact envelope. Yeah. Does that make sense? Okay. And hopefully, when we go through some of the, in more detail, some of the replication cycles, we'll see the story repeated, and hopefully that'll make a little bit more sense. Yeah, but these are great questions, you guys. Really, really good questions. Okay, and then folks, we probably won't be talking about these other viruses, maybe only in passing, but again, you guys, thank heavens, Ebola is an envelope virus. MERS and SARS envelope, coronaviruses are envelopes. Dengue and chikungunya, these are mosquito-borne. Thank heavens they're envelope, because that means again, they won't remain infectious in the environment for long. The only, like, except if you guys, we have moist organic matter in which the envelope viruses are contained, that moist organic matter, like feces, like vomitus, like tissue, right? Or maybe a lot of blood, as long as the virus is remaining moist within that organic material, that moist organic material is a protective form, right? So that's, you gotta be careful of that. Yeah?
You said that Ebola's overlooked.
RNA virus?
Yeah. But you know how they say that you get it from monkeys and bats, the meat?
Right. So it's probably, it's not that after the bush meat, like the wild animal meat that's containing Ebola virus, it's not after it's been cooked. It's in the preparation of slaughtering, butchering, preparing the raw meat that you're gonna get contaminated.
What if you dry the meat?
If you dry the meat, then that should make it safe. The concern would be if in the process of cutting up the meat to dry it, that do you contaminate yourself at that time? Yeah, so whenever you're working with, like, whenever you're butchering or slaughtering, you're always gonna be at high risk for infection, even with the envelope viruses, because, you know, there's a chance of nicking yourself or doing this, or, you know, contaminating your fingers. So that's always a concern, yeah. And what makes Ebola so hard is, in many traditions, the body of a deceased loved one is prepared extensively, and so they're seeing spread of Ebola from, so sad, like a family member dies and you're preparing the body for the funeral rites, right? And then you get infected, right? So this is really tough.
Is the disease
Very tough, yeah.
Sorry? Papua New Guinea that we.
Oh, okay, that is the kuru, so that's something different, that's called mortuary cannibalism, where the body is prepared and eaten, but it's the same thing, the women preparing the body of the deceased for a person, they're gonna get nicked and exposed to the kuru prion, yeah, so preparing bodies for funeral rites is actually pretty hazardous when you're talking about infectious disease agents, yeah. Okay, good questions, you guys, excellent. Okay, we're gonna keep going. And you guys, this is just kind of a repetition. So you guys, do you think this is naked or an envelope virus? Naked, right? And so what is this called? The capsid. It's made out of protein. Okay, and you guys, what do you think this is? The nucleic acid, right? So remember, you guys, the description of a virus is trouble wrapped in protein. Okay. So you guys, where would the adhesins be on this virus? On the capsid, on the outside. Good job, you guys. And again, folks, we're just emphasizing these naked viruses can remain infectious for long periods of time outside of their host. These are just some examples, you guys, of naked viruses. These are polio victims. So what happens in polio is the muscles that are involved in, like, walking and breathing become paralyzed. So back before we had the polio vaccines, they have these wards of people that were surviving what they called iron lungs. So the iron lungs, you guys go through cycles of positive pressure, negative pressure to help the people passively breathe. But I just can't imagine how horrible that would be. And then, again, you guys will be also later talking about a naked DNA virus, the human papillomavirus, the so-called wart virus. And this is one, you guys, we often don't talk about it, but this is rhinovirus, one of the so-called cold viruses. So we think of colds as being relatively mild upper respiratory tract infections. But just imagine, you guys, if I had a cold, an infection with rhinovirus, and I'm sneezing, achoo, you know, and coughing, and then I'm going, right? So remember, it's a naked virus, so it will remain infectious on the door handle for a long period of time. So if you were to touch it and then maybe go like this, you know, or I you know wouldn't do that, but you know, you could inoculate your mucous membranes and then you're infected. And then this is an example, folks. So here's our capsid with our nucleic acid. What's this on the outside? That's the envelope. Where did it come from? Stolen host cell membrane. And so what are all these, like, they look like lollipops? What are those? Those are the viral proteins, right? And we would presume, you guys, at least one of these viral proteins on the envelope would be the adhesion. And again, folks, remember, the envelope, very delicate, like olive oil, right? So anything that damages the envelope, the adhesives can't function anymore, right? And just a couple of envelope viruses we'll be talking about, you guys. Herpes viruses, and there's a whole family of herpes viruses. We'll talk about a few. And then this is electron micrograph of HIV escaping from a host cell. And again, you guys, just the best news that HIV is envelope, not gonna remain infectious for long outside of the host. Now, folks, I don't want you to panic. This is a table of DNA virus families, and what I'm really interested in is that we count how many DNA virus families there are. And just so you can interpret this, you guys, I inserted an N if the DNA virus family is naked. If there's no N, it means they're enveloped, okay? But right now, you guys, we just want to count the DNA virus families. 1, 2, 3, 4, 5, 6, 7. Okay, so 7 DNA virus families. Now let's look at RNA virus families. Wow. How many? 1, 2, 3, 4, 5, 6, 7, 8, 10, 11, 12, 13, 14, 15. More than twice as many. Now, as microbiologists, what would be your hypothesis? Why do we have more than twice as many RNA virus families? Remember, we talked about that high mutation rate? So that means there's going to be a lot more genetic diversity amongst the RNA viruses, right? So they branched off into more families, right? That high mutation rate means they're going to be really genetically diverse. Okay, guys, so what we're going to do, and sometimes folks feel this is not, it's like, well, why do we have to learn this? Because what we're going to do, you guys, is probably take almost the rest of the lecture talking about how bacterial viruses, bacteriophage, how they replicate. And so I hope you guys next week, when we start talking about how human viruses replicate, you're going to be blown away to see how understanding bacteriophage replication builds a foundation to understand how human viruses replicate. And historically, you guys, this is where we really first learn about viral replication. And the reason is, you guys, what must a virus have to replicate? To make copies of itself? Has to have a host cell, right? So if we wanted to study human viruses in the old days, you guys, what would we have to do? Purposely inoculate a human, right? And there's some ethical and moral issues with that, although some horrible things were done in the past, hopefully we've learned, right? But you guys, what kind of host cell do bacteriophage use? What's the host of a bacteriophage? Bacteria, right? And does anybody care if bacteria are killed? Yeah, right? So, the early work was done understanding viral replication using bacteriophage. The other thing, you guys, that's an advantage is the bacteriophage we're going to talk about, where some of the early work was done. Their host is E. coli. And you guys remember how fast E. coli can replicate? 20 minutes it can replicate, right? We can grow it easily. Doesn't take fancy medium, doesn't take fancy equipment. So E. coli is really a wonderful host to do any kind of study. So we're going to use bacteriophage and E. coli to explore viral replication. And then next week we'll take a look at human virus replication. Okay, so you guys might recall the earlier electron micrograph of one of the T bacteriophage and how amazing they are. They're just amazing. Oh, and look, a student gave me my own bacteriophage. It's got cute eyes. Well, they don't have eyes, but anyway. Okay, so you guys remember the bacteriophage that we're going to talk about, the T evenphage and the lambdaphage. They're what we call complex viruses because they just don't have like a capsid. So here you guys will see the viral capsid or head contains the DNA, and these two bacteriophage are DNA viruses. But then in addition, you guys, they have this incredibly elaborate tail. And this tail, this portion of the tail, you guys, it functions as a hypodermic syringe and needle. It's just phenomenal, and we'll see it at work here in a bit. And then these gorgeous tail fibers, you guys, these are these long tail fibers. We see these with the T even bacteriophage. is at the tip of the fibers of the special proteins, the adhesins will be located that will bind to specific molecules on the surface of the bacterial cell wall, okay? And then we'll see we have a base plate with these little spikes that will play an important role, okay? So for what we're first gonna do, then we'll set the stage here. There's two different ways that bacteriophage, bacterial viruses can replicate. And the two different types of replication cycles, you guys, are called lytic replication and lysogenic replication. So let's first talk about lytic replication, which is the most common type of replication. Okay, so again, you guys, and yesterday I confused folks because I'm using 3 different terms to describe bacterial viruses. So bacterial viruses are also called bacteriophage. And we're bad, you guys, we start shortening their name. A lot of times you just call them phage. So when we're talking viruses, if you hear phage, you automatically know it's a virus of bacteria. Okay, all right. So folks, as we said, there's gonna be 2 bacteriophage replication cycles. So we'll do the simplest one first. So replication means to make copies. Okay. So the first one we'll describe, you guys, is called the lytic replication cycle. Lytic because it always results in lysis of the host bacterium. So the Lily coli is going to die in this one. And our example, you guys, here is a so-called lytic phage, the so-called T-even bacteriophage. All right. So you guys, what we'll do is we'll go through the steps and then we'll look at the cartoon, okay? So step 1, you guys, in any type of viral infection is the virus has to attach to the host cell. So in this case, it would be attaching to the bacteria. Phage adhesins are going to bind to specific receptors on the bacterial cell wall. Then you guys, the phage is literally going to inject the phage DNA into the bacterium, so that's called entry. And then once the DNA gets in there, that bacterium's a goner. And then the phage DNA is going to be transcribed by the bacterial RNA polymerase, and then the phage mRNA will be translated by the bacterial ribosomes into phage proteins. That's called the synthesis phase, where the poor little E. coli, now all it's doing is making phage DNA and phage proteins. And then in this wild event, you guys, the phage proteins assemble into capsids. So you guys, let's pretend we're inside the E. coli. So the capsomeres are going to assemble, and the capsid, the heads will start to self-assemble. This is just like, to me, the wildest science fiction. And you guys, what's wild is then the capsids then will grab pieces of DNA from the cytoplasm and package the DNA inside the capsid head. Is that not the wildest thing? Okay? So now we have our capsids, our heads are packaged with the phage DNA, and then the tail proteins assemble and become attached to the capsid, the head. So we end up with our assembled bacteriophage, right? Self-assembly, that's just crazy. And then once the phage are assembled inside the bacterium, they release a lysozyme-like substance that helps break down the peptidoglycan. And the poor little bacterium is going to burst open, it's going to lyse, releasing all these newly replicated phage that then are going to attach the neighboring bacteria and start the cycle all over again. Okay, so let's take a look at the cartoon of this, folks. So here's a lytic replication. So step 1, you guys, always attachment. So folks, just so you know, here's our E. coli, here's our bacteriophage. The E. coli chromosome is in purple, and the phage DNA is kind of turquoise. So we have attachment, and then you guys, and we'll see more detail of this, the phage contracts and injects the bacterium with the phage DNA. The phage DNA, as we said, will be transcribed and translated, and as a result, The bacterial chromosome is gonna be broken down to act as a source of nucleotides to make more copies of the phage DNA. So here, we're in our synthesis stage here, we have the phage DNA replicated. Phage proteins are assembling into capsids and tails. The capsids package themselves with the phage DNA, I just think that's so bizarre. The tails and the capsids assemble. The release of the peptidoglycan causes lysis of the bacterium, and all these replicated phage are released. Okay.
So does the bacteriophage DNA or RNA have some sort of enzyme that breaks down the bacterial, that breaks it down?
Right, that's right, that is
So
right.
like a gyrase or a helicase or something?
I would call it a general nuclease,
Okay.
to tear apart the bacterial chromosome, and that's right, because that's gonna be the source of nucleotides to make copies of the phage DNA.
So is that part of the bacteriophage DNA and RNA?
Yes, yes. And there's a group of genes called the lytic cycle genes, so all the phage genes associated with the lytic cycle, like all those nucleases, would be part of this lytic cycle. Those genes would be expressed during the lytic cycle. Yeah. You know, poor little E. coli. Does all that work? It's just so, anyway, all right, can't get sentimental, right? You guys, it's just a bacterium. So you guys, this is, to me, is just the wildest sequence of events. So this is the first step to getting attachment, and just to get you oriented, here's a bacterial cell membrane. This is the inside of the bacterium. It's E. coli, right, gram-negative, so thin layer of peptidoglycan. What's this on the outside, you guys? Outer membrane, yeah, okay. So the phage attaches, and now watch this. It contracts the tail, you guys, and drives this shaft across the cell wall, across the cell membrane into the cytoplasm of the bacterium. And during that contraction, it drives the DNA into the bacterium. So you guys, this is crazy making. So the tail is acting like a hypodermic syringe needle. I mean, it's not even a cell, and it's so complicated. To me, it's just mind blowing, right? So once that phage DNA gets into the bacterium, that bacterium is going to be a goner. This is just showing that wild assembly process, you guys. So we talked about how the capsids will grab pieces of DNA from the cytoplasm and package them. During assembly, most of the DNA in the cytoplasm will be phage DNA, but occasionally you guys will see there's a little mistake made. A little piece of bacterial chromosomal DNA might be grabbed and packaged instead. And we're going to see, you guys, that's how we get what's called generalized transduction, and we'll come back to that. And then, you guys, here we're seeing assembly of the tail with the capsids and the tail fibers, and here we have our mature bacteriophage. All right, you guys, now that was the easy one, right? Now we're gonna do the hard one. So the more complicated cycle, you guys, is called the lysogenic cycle. So lysogenic replication. And only a subgroup of bacteriophage can carry out lysogenic replication. This is very kind of fancy if you get to do this. So the group of phage here, you guys, they're called temperate phage. Only temperate phage. And our example, you guys, is going to be lambda. We use the lambda symbol, phage of E. coli. And what we're going to see, you guys, in the lysogenic cycle is the E. coli will not be killed, right? And so I think of like temperate. Somebody has a temperate personality. They aren't angry, or they're not nervous. They're mellow, kind of nice to get along with. So that's why I think of temperate, right? The bacteriophage won't hurt the E. coli if that's of any help. So you guys, just as a heads up, on lecture exam 3, there'll be lots of compare and contrast type questions, like compare and contrast lytic to lysogenic replication cycle, compare and contrast lytic bacteriophage to temperate bacteriophage. So just so you know why I'm asking some of these questions, we're doing compare and contrast, okay? So you guys, if we were comparing and contrasting lytic replication to lysogenic replication step by step, we'll see in lysogenic cycle, you guys, the first 2 steps are the same as in the lytic cycle. we have attachment. And you guys, with lambdaphage, they too are complex bacteriophage. They have the protein capsid, the head with the phage DNA. They have the tail, the helical tail here, but they lack the big, long, beautiful tail fibers, so they aren't quite as elegant. But basically, they function the same way. So folks, lysogenic cycle, the phage attaches, right? It injects its phage DNA into the bacterium, so the first two steps are the same as lytic, but it's the third step, you guys, where things go really differently. So instead of the phage DNA being transcribed and translated and basically initiating the death of the E. coli, instead, you guys, this is so wild, the phage DNA inserts itself where? Into the chromosome. Isn't that wild? So you guys, so here's special vocabulary. So you guys recall the phage inserted into the bacterial chromosome. We call it a prophage. Okay, so you guys, if I ask you what is a prophage, what would you tell me? Phage DNA inserted into the bacterial chromosome. That's a prophage. And you guys, we would call the bacterium that carries the prophage we might call it a bacterial lysogen, or we could call it a lysogenized bacterium. So folks, if I ask you what is a lysogenized bacterium, what would you tell me? It's a bacterium carrying a prophage. And what's a prophage? Phage DNA inserted into the bacterial chromosome. Okay, you guys all right with that? Okay, and the coolest thing is, you guys, is now the prophage, it simply becomes part of the bacterial chromosome. When the bacterium is ready to divide, it has to copy its chromosomal DNA, right? So do you think the bacterial DNA polymerase will copy the prophage right along with the rest of the chromosome? Oh yeah, I mean, DNA polymerase, it's like it's DNA, right? So you guys, so the prophage can be passed down generation after generation after generation, right? Many, many, many generations can inherit the prophage, right? Which is kind of cool. And we'll come back and try to explain why we think this might be of advantage. But you guys, something wild can happen. Something really wild can happen. And that is the phenomenon of induction. And induction, guys, another vocabulary word, right? Induction is when the bacteriophage switches from the lysogenic cycle back to the lytic cycle. Okay, so induction is, we're in the lysogenic cycle. Induction is when we switch from lysogenic to the lytic cycle. Okay, and it's usually triggered by DNA damage. DNA damage triggers induction. Okay, and you guys, this gets really thick. So let me see if, okay, let me just advance one, you guys. Okay, sorry. So before we hit induction, you guys, this is just a description of lysogenic cycle. So remember, the first two steps are the same as lytic attachment and entry. It's the third step that's different, you guys. So remember, in the third step, we have formation of the prophage. The bacterium carrying the prophage is called a lysogen or lysogenized bacterium. The prophage will be replicated as part of the bacterial chromosome and passed on generation after generation. Okay? And you guys, this is a key actually to understand induction. It turns out that the temperate phage, lambdaphage, they have a choice when they infect the bacterium. They can either do the lytic cycle or the lysogenic cycle. And we'll come back and put this on the board, you guys. So the question is, if lambdaphage can do lytic or lysogenic, what determines which one it's gonna, which cycle it will use? And it turns out the key, you guys, is a phage repressor protein right? So there's a phage repressor protein that will block transcription of the lytic cycle genes. So if in the newly infected bacterium, if there's sufficient phage repressor protein made, it'll block the genes for the lytic cycle, and it automatically sends the bacterium into the lysogenic cycle. Okay? Now I know this is like, what the heck are you talking about? So what happens, you guys, in induction, right? We're going to switch from the lysogenic cycle to the lytic cycle. And you guys remember how we said it's usually triggered by DNA damage? Well, in the complicated series of events that happens in a bacterium when its DNA is damaged, there will be proteases that end up destroying the phage repressor protein. And that's what permits the phage to switch from the lysogenic cycle to the lytic cycle. So let's just write that up here, you guys. So let's say DNA damage triggers induction, and this is destruction of the phage repressor protein. That's the molecular event that permits transcription and translation of the lytic cycle genes. Okay. So in induction, you guys, it's absolutely wild. The prophage cuts itself out of the bacterial chromosome and then enters into the lytic cycle. So we're going to get the synthesis stage, assembly, and then lysis of the bacterium. It's just wild. Okay. And the result will be the release of new phage with lysis of the bacterium. Now, the next slide, you guys, it's a cartoon, and to me, it's always cartoons are much easier to understand. Okay, so you guys, we'll walk through this. So here we have our lambda phage attaching, injecting, and we're going to presume, you guys, there's lots of phage repressor protein, so it's going into which cycle? The lysogenic cycle, right? And again, you guys, once the phage is inserted in the bacterial chromosome, it can get copied and passed down generation after generation until what happens? DNA damage. And you guys, next week we'll be doing lots of DNA damage to our bacteria. UV light is a great way to cause DNA damage. Remember those tiny dimers? Yeah. So for example, you guys, if this E. coli, this lysogenized E. coli was exposed to high levels of UV light causing DNA damage, the phage repressor protein is destroyed. It triggers expression, transcription, translation of the lytic cycle genes. The prophage, you guys, does what? It cuts itself out of the chromosome, right? And here we're going to enter into the lytic cycle, where we're going to have synthesis, assembly, and then release. Now, it's like, for me, I need a story, right? I can't remember things unless I tell a story. So you guys, my analogy is that the bacterium is a ship out on the ocean, and the prophage is a rat on the ship. And the rat's really happy. It's got a lot of food, it's got a warm, dry place to live, right? Until one night, the ship hits a iceberg. Coral reef, right? Something really bad. And what's happening to the ship? It's sinking, right? What's going to happen to the rat? It's going to die, right? Unless it jumps ship. Get out of your sinking boat. Get out of your dying bacterium. Does that make sense? So the way the phage rat jumps ship is via induction. It cuts itself out of the chromosome, quickly triggers that lytic cycle, makes copies of the phage DNA, gets it packaged into, right, the protective phage heads, and then escape before, well, they're helping the ship sink, right, by enticing the bacterium, maybe putting the poor bacterium out of its misery, right? So you guys, does that make sense? Yeah, no.
Does that still happen after, so right, if you're in the lysogenic replication cycle, the daughter cells of the original bacteria have
The repressor protein, yeah.
but they have the
Capacity.
prophage in them still?
Yes. If you're in the lysogenic cycle, the lysogenized bacterium, every time it divides into the so if this, like this E. coli was a pathogen,
But does it make
could the
a difference?
lysogenize, oh, thank you for asking that. Okay, so you guys, the question is, if I understand, could the a lysogenized E. coli behave differently if it infected, say, a human, compared to the non-lysogenized bacterium? And to answer that, Martin, we need to go to the next topic, which is specialized transduction. So short answer, yes, because these prophage can carry powerful toxin genes that can end up killing us. Whereas the E. coli not carrying the prophage wouldn't have the toxin gene. It would maybe be like a good member of our intestinal tract. But these lysogenized bacterium, because the prophage can carry toxin genes with it, lysogeny might take a non-pathogenic beneficial bacterium and turn it into a horrible pathogen that could kill us. And you guys, this is perfect because the example we're going to use is the evolution of E. coli 157H7, the Shiga toxin producing E. coli. And just to cut to the chase, you guys, E. coli 157H7, that Shiga toxin producing strain of E. coli, it's lysogenized, it's carrying a prophage, and the prophage is who brought the Shiga toxin gene there on board. If that E. coli didn't have the prophage, it would probably be a member of our good beneficial microbiome. But because that prophage with that toxin gene is in the bacterial chromosome, it can kill us. Yeah, so excellent, excellent question. So you guys, this one, yesterday I did such a terrible job on this one, and the students were moaning and groaning. Because what we want to do now, you guys, is talk about, going back, we want to talk about one of the types of horizontal gene transfer that involve bacteriophages. You guys remember transduction? Yeah. So you guys remember how we said there's two different types of transduction? And we said we aren't going to talk about the second type until we do this. Well, then we get to talk about generalized versus specialized transduction. And are your antennae going up saying like, this sounds a bit like this could be a potential compare and contrast exam question? Yeah, okay, so your antenna should be fluttering, you guys. Okay. Now I hope I'll do a better job this time around, you guys. Okay. So folks, let's talk about generalized transduction. In generalized transduction, any donor bacterial genes can be transferred to recipient bacterium. So in general, any donor bacterial chromosomal genes can be transferred to a recipient. So let's just walk through this, you guys, to see how this would work. So in generalized transduction, you guys, we have to have a lytic cycle. Okay, so you guys, this is going to be our donor bacterium, right? It's going to be infected by the phage, attachment, entry, biosynthesis, assembly, and release, right? But you guys remember during assembly, remember how we said the capsids are just randomly grabbing pieces of DNA from the cytoplasm? Now most of the DNA, you guys, will be what? Phage, right? Phage DNA. But remember you guys how we said the chromosome is going to get cut up here? There can still be little fragments of donor DNA in the cytoplasm. And the capsid can't tell the difference, so occasionally a mistake is made in what gets packaged. Donor bacterial DNA, right? And what's wild, you guys, is that phage that we call a defective phage, because it's not carrying phage DNA, it's still infectious, right? So what would happen, you guys, if we could pretend, I wish I had a little marker here and might make this purple. Let's say this is a defective phage that packaged a piece of donor bacterial chromosomal DNA. When it's released, it's still infectious. So that it can bind to a neighboring bacterium and inject it with that little piece of donor chromosomal DNA. And if there's homologous recombination, that donor DNA will get inserted into the recipient's chromosome. Right? So the reason this is called generalized transduction, guys, is any piece of the donor chromosome could be transferred to the recipient. So in general, any piece. Does that make sense? Okay, all you need is a lytic cycle. So either the lytic phage, the T-even phage can do generalized transduction, or you guys remember, the temperate phage, they can do lytic too. So they can also carry out generalized. The hard one, you guys, oh my goodness. If you're about to fall asleep, this will do it to you. Okay, the harder one to explain, you guys, is specialized transduction. And let me me see you guys if this, I was having a hard time finding a nice cartoon of this. Okay, this one is really not too bad here, okay, so you guys, how about this? Let's see if we can make this one work. So you guys, in specialized transduction, you have to have both the lysogenic cycle and the lytic cycle, and therefore, you guys, which group of bacteriophage can carry out specialized transduction? Only temperate, right? Because temperate phage carry out both lysogenic and lytic. Our lytic phage, you guys, the T-even phage, all they do is lytic. They can't do lysogenic, therefore they can't do specialized transduction. So we're talking here, you guys, temperate phage. So you guys, let's see if I cannot murder this. So here's our donor, bacterium. Here's our lambda phage. And we're going into what's part of phage replication, you guys? The phage DNA is inserted into the chromosome, so this must be this is a lysogenic cycle, right? Okay. Now, you guys, let's say we have induction here, so some kind of DNA damage. So induction, what do we know about induction? The phage switches from lysogenic to lytic, right? Now, in specialized transduction, what happens is, in induction, when the phage is trying to cut itself out of the chromosome, it makes a mistake, and it cuts into the donor chromosome. So when the phage cuts itself out of the chromosome, it takes a little piece of donor DNA with it. And special donor DNA, you guys, special because it's going to be donor DNA on either side of where the phage inserted, right? So only special genes are going to be transferred to the recipient. And what happens, you guys, is this becomes the template, this combination of phage and donor DNA, that becomes a template for phage replication during synthesis. So all of the phage DNA that gets copied will have phage DNA and those special donor genes. And so subsequently, you guys, all of the phage, when they're assembled, all of the phage are going to be recombinant. They're going to carry phage and donor DNA. When they're released, they're infectious, right? So here's the recombinant phage, you guys, binding to the bacterial recipient, which luckily has light blue chromosomal DNA, right? So we have the phage and donor DNA injected, and the phage donor, excuse me, the phage and donor DNA, you guys, it can insert into the chromosome, or in some events, you might just get the donor chromosomal DNA inserted via homologous recombination, right? So to me, it's a little bit similar to say both phage and donor DNA will be inserted into the chromosome of the recipient. So again, you guys, this is called specialized because only the special genes on either side of where the phage inserted itself will be transferred. Now again, you guys, it's like, oh my God, it makes my head hurt. Why do we care? Well, you guys, this is exactly how E. coli 0157H7 acquired the Shiga toxin gene. So let's see if we can't cartoon that, you guys, just very crudely. As an example of specialized transduction, and exactly what Martin was saying, well, are these lysogenized bacterium, are they more dangerous? It's like they can be really dangerous. Okay, so this is an example, this is a specific example of why we're so interested and worried about. So this is an example of specialized transduction. And this is the acquisition of the Shiga toxin gene by E. coli 0157H7. And in lab this morning, the morning folks, and afternoon, you guys, we're gonna discover this is what we call the serotype. It's a way for us to identify specific strains of pathogens. Very important. Okay. So folks, you might remember. We did a little bit of the Shiga toxin story, you guys. So remember the original. This is going to be our donor bacterium, and it's a cousin of E. coli called Shigella dysenteria. Causes bacillary dysentery. And as far as I know, this is the source of the original Shiga toxin gene. So I'll just put it like right here, you guys. So the story goes that somewhere, at some point, the Shigella dysenteria was infected by a temperate phage, and guess where the phage DNA inserted itself? We'll put it right about here, you guys, right? So this is gonna be phage DNA. So we had a Shigella dysenteria that was lysogenized and was carrying a prophage right next to the Shiga toxin gene. And then the hypothesis is there was induction. And what happened was when the phage cut itself out, it made a mistake and did what? It also cut out the Shiga toxin gene, okay? So following induction then, this is in the lytic cycle, right? So we're gonna go from lysogeny to the lytic cycle. And so the template, you guys, for the phage DNA replication, it was all this recombinant. So it was phage DNA, and what was also part of that template, you guys? The Shiga toxin gene, right? The donor DNA. So that became the template. So during biosynthesis, all the phage DNA was carrying the Shiga toxin gene as well. And then this, of course, is what was packaged during assembly, right? And you guys can all just put assembly here. So all of the newly replicated phage, they're all going to be recombinants, right? Because what are they all carrying? Phage DNA and what? And, yeah, exactly, guys. They're all carrying the Shiga toxin gene. So you guys, what do you think happened next? Who do you think that recombinant phage infected? Yeah, exactly, guys. That's exactly what happened. So then these recombinant phage remember, they're still infectious, right? So these recombinant phage were able to infect a close cousin of Shigella, right? E. coli and Shigella are a close cousin, and inject it with what? Oh gosh, now I'm gonna get in trouble learning out of colors here. Yeah, so let's say this is the recipient, this is our recipient. This is the recipient chromosome. Okay, so when the recombinant phage injected the recombinant DNA, so there's our phage DNA and there's our Shiga toxin gene, what happened, do you think? What do you think happened? Yeah, in this case, you guys, both the phage and the Shiga toxin gene inserted in the E. coli chromosome. And the reason we know that, they do DNA sequencing, and they're reading E. coli chromosome, and boom, all of a sudden they hit phage DNA, and what's right next to it? The Shiga toxin gene, right? So they're able to prove this, right? That it was through specialized transduction. So you guys, that's how the E. coli 0157H7 acquired the Shiga toxin gene. Okay, and before I forget this, you guys, because this was the original story I learned about Shiga toxin producing E. coli. And so a few years ago, there was an outbreak of Shiga toxin producing E. coli in Europe, but they had a totally different serotype. Like, it's one in the lab exam study guide. Yeah, exactly, the 0104H4. I was like, wait, this isn't E. coli 015787. But you guys, that's silly for us to think that this can only happen with one E. coli, right? Because there's many, many different types of E. coli, and it's possible for this event to happen to many other strains of E. coli. And what I don't know, you guys, but I think it would be possible, could this guy, our E. coli 0157H7, now act as a donor, right? Does that make sense? If we had induction here, right? The phage, Shiga toxin gene, you get cut out, packaged, and maybe another strain of E. coli gets infected. So you guys, for me, it's like, I was like, oh, so the Shiga toxin gene can be shared amongst multiple strains of E. coli. The reason, you guys, I make a big deal about E. coli 0157H7, this is probably the most infamous Shiga toxin producing strain of E. coli in California, right? So this is probably like the most famous one. So folks, not to make you crazy, but to me this is really important that you know about the Shiga toxin and its effects. Have we talked about what kind of damage Shiga toxin does? It's really important here in California. You guys just yell at me if we've done this already. Because I have noticed as I age, I tend to repeat myself. Okay, so you guys, the Shiga toxin, okay, so we presume, you guys, that, do we know what the transmission is of E. coli 0157H7? Yeah. So it lives in the intestine, right? So how will it be shed? In feces, right? So you guys, what would be transmission then? Fecal oral, right? So wherever the feces goes, the E. coli 0157H7 goes. And what's tough about this, you guys, is that humans and non-human animals can be colonized. Okay, so feces, and then so we have fecal oral transmission. Wherever the feces goes, it contaminates food or drinking water, and we consume it. That's how we would get colonized. Now, the reservoir, did we talk about the reservoir, you guys, like here in California? Cattle, right? So cattle are thought to be important reservoirs because their cells lack the receptor for the Shiga toxin. So cattle can be colonized and not have clinical signs and symptoms. I've read sometimes maybe baby calves, sometimes will have a diarrhea, but generally if you have an adult cow or bull or steer, you can't tell they're infected, right? But their feces is chock full of E. coli 0157H7. So initially, you guys, this was called the hamburger disease. And the hamburger disease, because, during slaughter, you guys, the animals sense something's wrong, they go to fight or flight, they lose control of their bowels, and so there's feces everywhere. And the folks doing the slaughtering, they're trying really hard to keep the carcass clean, but it's hard, you guys. Once you take the hide off, the skin off, it's hard not to get some kind of fecal contamination, right? So the idea here, you guys, the reason it was initially called the hamburger disease, and let's think about this whole process of fecal contamination of the cattle carcass. So you guys, let's pretend this is the cattle carcass after the hide's been removed. So you guys, where would the fecal contamination be? On the big carcass? It'll be on the outside. Luckily, you guys, these, like E. coli, it's not tissue invasive, right? So the fecal contamination will be on the outside, right? So we would presume if E. coli is present, right, it's going to be on the outside. So now let's think, you guys, how this carcass will be prepared for human consumption. So you guys, so we might have big solid pieces of beef, say a steak, right? A solid piece of meat. Where will the fecal contamination be? On the outside, right? Because remember, the E. coli, it doesn't invade the tissues. It's going to be part of the surface fecal contamination, right? So for example, our E. coli or any other fecal pathogen is going to be on the outside. So you guys, we can argue, since E. coli are heat sensitive, if we had a solid piece of meat like a steak, and this is a microbiology barbecue, right? We could argue if we made sure to sear the outside, sear the surface, that will kill any fecal pathogen present. And in theory, you guys, as long as you're searing the outside where the fecal pathogens are present, you could probably more safely eat a solid piece of meat, a steak, say medium rare or rare, not cooked all the way through. But you guys, let's take that steak, or more likely the trimmings of the carcass, maybe not the best parts of the carcass, right? And we grind it up to make ground beef, hamburger, right? Make a nice hamburger patty. So you guys, where will the fecal contamination be now?
Everywhere.
Everywhere, right? You grind in the fecal contamination. So you're gonna have fecal contamination in the middle of your hamburger patty. And who else could be there? Any fecal pathogen, including E. coli, 015787. So fecal contamination throughout, in any kind of ground meat, you guys. It could be ground turkey, ground chicken, anything where you grind it together, fecal contamination will get spread throughout. So you guys, so let's do the microbiology party barbecue. And you say, oh, I want my hamburger rare. Why is that not a good idea? Yeah, so you guys, ground meat, ground hamburger, make sure it's well done, right? Because if it's not well done, thoroughly cooked, those fecal pathogens, if there's some present, will still be alive in the middle. And you guys, this had heartbreaking results. Because this was the story of E. coli 157H7. It was called the hamburger disease because it was associated with what? Yeah, undercooked hamburgers. And what I remember, you guys, was the Jack in the Box outbreak. And remember, you guys, Jack in the Box, American culture, we're famous for fast food. Well, sometimes it's too fast. It's not cooked, right? And this was really sad, you guys, because what happened, and they're still not sure if it was because the hamburgers, the Jack in the Box hamburgers, they're not sure if it was, were they improperly cooked, or after cooking, did they get contaminated, right? So, you guys probably know this, but like, if you have, say, hamburgers on a plate, and say you put them on your barbecue, you cook them, I mean, I have seen this, then you take the cooked hamburgers and put them back on the same plate the raw hamburger was on, right? So that's not a good idea. The same thing with cutting boards, you guys, right? If you're using a cutting board maybe to make your hamburger patties, and then after cooking you put them there, right? Probably not a good idea. So they're not sure. Did the hamburgers get contaminated afterwards? They're not sure. But I think at least one little child died from this, you guys. Little kids are very susceptible. It's just heartbreaking, right? And then, you guys, what's wild is over the last maybe couple of decades, now we're also seeing it in fruits and vegetables. Another horrible outbreak, you guys, was in the old days, on Wallen, the juice company, really a healthy juice, right? So in the old days, they would not pasteurize their fruit juices, right? Because again, a lot of people think it's more nutritious if you don't pasteurize. Well, they had apples. And they made unpasteurized apple juice from it. And guess what that unpasteurized apple juice had in it? E. coli 157E7. So you guys, again, they don't know how it happened. They don't know if, like, there was animal feces in the orchard. Did some of the apples fall into the feces and get contaminated? Although you're never supposed to use fruit that's dropped on the ground to make like an unpasteurized juice, so they're not sure if it was that. You guys, we, adult humans, we can act as a reservoir, so it's just not cattle, humans. We can act as reservoirs, so any of us that are handling the fruits or vegetables, if we have E. coli 157 and we've got contamination of our fingers, we can contaminate the fruits or vegetables. And the reason fruits and vegetables, you guys, very often we don't cook them, right? So if we cook them, no problem. But if we're eating the fruits raw, right? Or we're eating the vegetables raw. And another example, you guys, was here in California a few years ago, we had a big outbreak associated with California spinach. And this was an interesting story, you guys. Huge outbreak. California, we ship produce everywhere, around the United States, around the world. So we had E. coli. California was the E. coli 157H7 capital of the world there for a while. So they're trying to figure out how the California spinach got contaminated. And when they went to look at the fields, there was a dairy nearby. So they're like, well, could the cattle feces with E. coli maybe contaminate the irrigation water? And that's how the spinach got contaminated. Was it human contamination, right? And what they finally discovered, you guys, it was wild pigs. So wild pigs had broken into the field and were having a lovely spinach salad dinner, and they were carrying E. coli 157H7. So when they defecated, they contaminated the spinach. And you guys, I love this stuff. Like the spinach, it comes in these pre-washed, quote unquote, bags. And these are the best little microbial incubators. You guys, so your bagged spinach, lettuce, the pre-washed, pre-bagged stuff is the best incubator for microbes. So anyway, the contaminated spinach was a source of E. coli. And I don't know if there were any deaths associated with that, you guys, but this is a nasty one. Yeah, so the bottom line is, you guys, with any kind of ground meat, especially if you're feeding it to a little child, somebody older, anybody with a compromised immune system, always make sure that meat is well done, right? Because it could be any fecal pathogen, right? And I always just kind of try to keep my ears and eyes peeled for outbreaks. And you guys, I love Trader Joe's. I mean, I adore Trader Joe's, but they have a number of food recalls. So just always, go up and check at the checkout stands. They'll always have the food recalls out there. So my thought is I should go and check the food recalls before I shop there, right? And you might say, why Trader Joe's? Because a lot of their prepackaged stuff, you guys, they're bringing in food from all these different sources, right? And the more different sources, the greater is the chance of contamination. Yeah, so you guys, I love Trader Joe's, but it's just like, just keep your eyes and ears open. Okay, so folks, just in the last little bit, and I know I'm kind of hammering this. Kristen, you have a question? Yeah.
Yeah, I just wanted to kind of go back over this really quick and make sure I'm getting it right.
Yeah, can I just finish one last bit, you guys, on clinical signs and symptoms of this, because it's important that we know how to identify it, especially in little kids. Okay, so you guys, the Shiga toxin, did I mention what its target is? So it shuts down, it inhibits ADS ribosome. Who has ADS ribosomes? We do, right? It's the eukaryotic cytoplasmic ribosome. So if we're colonized in the intestine, some of the clinical signs you see, you guys, is a bloody diarrhea. Not always, but in a weird sort of way, you guys, if you have children and they get colonized, I would almost hope they get a bloody diarrhea, because that's an early sign you need urgent care, right? So if you take them to the ER, you might need to insist that your child is screened for a Shiga toxin producing E. coli, because my understanding is it won't automatically happen, which to me is just like, are you crazy? I'm okay with bloody diarrhea, right? And then in some folks, you guys, it gets worse. Because the toxin is absorbed and it causes blood vessel damage. And as a result, your blood cells, your erythrocytes, start to lyse. Right? So that causes RBC lysis. And that results, you guys, in a hemolytic. What does hemolytic mean? Red blood cells lysing, right? Erythrocytes lysing, anemia. Right? So you don't have enough red blood cells. You're not able to deliver sufficient quantities of oxygen to your cells and tissues, right? And then this is like, and then, right? And then in some cases, the toxin reaches the kidneys and causes kidney damage. And a sign of kidney damage, you guys, is you get increased urea in the blood. And that's called uremia. So folks, when you have multiple problems going on at the same time, you call it a syndrome. So Shiga toxin can cause this life-threatening syndrome called HUS. What do you think that stands for? Hemolytic? What's a U for? Uremic syndrome. And you guys, this can kill. And they've even seen in folks that develop HUS, they survive. They might have lifelong kidney damage, right? So this is just a nasty one. And we've got it here in California, right? So just be aware, you guys, just be aware. And childhood diarrhea, you guys, I would consider that a medical emergency, and I don't care what they tell you. It's like you need to have them screened for Shiga toxin producing E. coli. So Kristen, sorry, yeah. So thanks, you guys. Thanks for being patient with me on this topic.
Just to recap, with the specialized strain transduction,
Yes.
lytic and lysogenic cycles. So
Yes.
the cycle switches from lysogenic back to lytic.
Correct.
The prophage picks up a special piece of the donor DNA,
Yes.
and then the prophage of the donor DNA will be transferred to a new host,
Correct.
and the recombinant DNA will be the new template.
Well, that would be in, if we back up a little bit, so induction, we're switching from lysogenic to lytic.
Right.
The prophage makes a mistake when it's cutting itself out of the chromosome, it cuts into the donor chromosome. So the template for synthesis is the recombinant phage donor DNA.
Okay.
So all of the phage that will be the product of that lytic cycle will all be recombinant phage.
Okay.
And then they inject the phage DNA with the donor DNA into the recipient. And in the case of E. coli 157H7, the phage DNA and donor DNA got inserted into the chromosome. Another possibility is you might only get the donor DNA through homologous recombination and get inserted into the chromosome. And you guys, I'll end with this, and I'm so glad that Kristen asked this. So, people remember how we're saying, why? Why is this going on? Right? So you guys, this is another way where the bacteria, which normally reproduce asexually, they are increasing genetic diversity. This is another way for bacteria to mix their genetic information. So remember, as biologists, we're like, there has to be a survival advantage to this, right? So we can argue with the bacteria that this transduction is a good thing because it helps generate more diversity, sharing genes, mixing genes, right? For the bacteriophage, you guys, it's win-win because we could say, and I know this sounds kind of harsh, but we could say as biologists, like the goal of life or even non-living things is to copy your genetic information and pass it on. So the phage, this is awesome. They have two different ways to copy their phage DNA, lytic or lysogenic. So it's win for them as well. Okay, you guys, I know you're so